And Assalamu alaikum wa rahmatullahi wa barakatuh. It's a pleasant morning today, and first of all, I like to thank the organizers, Orion and Vance, for having me here to present some of my work. And just to make myself relax, I just want to give you the brief of how I started my research on cancer. So my boss first brought ascitic tumors from France to cultivate them in India so that I can begin to work. He has a group where I was the participant and we worked on ascitic tumors. That was my first interaction with cancer tumors. So starting from there, then we didn't have much facilities to do high-end research, but we did all the macromolecular profiling of those cells, and then we published a lot of papers that RNAs, concentrations of RNAs and expression is more than any other macromolecule and all. So my work stopped after that because I came back to IICT on a promotion, and where I was the deputy director there, and I established a toxicological lab. That was the demand of the job. And then I went back to research as emeritus scientist in Indo-American Cancer Institute, where I continued to work on cancer. And there I had the opportunity to look at biopsies of human cancer cells, and then I had the opportunity to do high-end research work in this. So we have been working on an area called pharmacogenomics, where we used to look at the drug and gene interactions. We could isolate lots of cancer genes which are listed. So many genes are listed, profiles and number of genes in cancer. Actually, cancer is due to this genetic instability. So the cell has lots of genes which are turned on and turned off in the wrong ways. So that is how it came into recognizing that these genes play a very important role in the process of carcinogenesis. So that is how we continue to do our research in this, and we help the oncologists to determine the phenotype and the genotype. That means the SNPs related to cancer, that means the mutations which you were talking about yesterday, those were the ones which are very important and gives us an idea about if a certain gene is turned on or turned off, then there is an influence on the therapeutic response to the person's cancer resistance or cancer susceptibility. So that is how it started off. So now coming back to my topic, which I will talk about today, is the latest trends in cancer therapeutics. What is this? It is about MicroRNAs, which are, of course, they are tiny. MicroRNAs means small RNA molecules. These are really non-coding RNAs, which they don't code any protein, like mRNA goes for making the protein, but they cannot code for any protein, but they interfere into the making of the proteins. So in the past decade, these have been extensively studied in a different of human cancers. So that's why my interest was to look at, can we identify these, because they have a dual role about tumor suppressor, also as tumorogenic, oncomeres. So here, as you know, that single nucleotide polymorphisms may also occur in these microRNAs, because they are very tiny and they can always, they don't pair up with their partners, then there is always a mutations and change in the SNP. However, the role of genetic variants in the microRNAs on cancer susceptibility, still it remains largely unknown. We still don't know if this happens, what is going to be the next this thing. So these are the kinds of pathways we need to unfold in this kind of research. So we have these microRNAs which are associated with intrinsic resistance also, and also they are both for tumor progression, and they also play a role in metastasis. So these are very important, and they are expressed in cancer to a greater potential, and identifying this will become cancer biomarkers. So if we know that which is this microRNA, which is expressed low or expressed high, or where is the SNP, then it becomes cancer biomarkers. So biomarkers are very important to identify, to tackle with the treatment procedures or for identification of disease diagnosis or prognosis. So therefore, almost 2,000 RNAs are known so far in human cancers, and they have a key regulatory roles in the development, differentiation, apoptosis of normal cells as well as in other phenotypes of cells in carcinogenic process. So here this is just the timeline of the discovery of the RNAs. See, Francis Crick, when he left Watson, then he joined Leslie Orgel and proposed that RNA was the first information molecule in the before 70s, around 70s. Then this Harry Noller proposed the role of ribosomal RNA in translation of these into proteins. And then we had the discovery of this botanical research scientists working on petunias to discover about the gene silencing technologies, because petunias are purple in color, white in color, and so many colors, right? So when they cross, then some colors are not expressed in the flower. So they discovered that there are silencing genes which could be silencing the color expression profiles. So that was the discovery of the gene silencing technology. So that's the same, because here also microRNAs are playing a role. And here we have the, next we have the Human Genome Project, which was initiated, and it began to increase exponentially when Craig, really, he unfolded the entire Human Genome Project before the turn of the century. Then we have the Go and Kempukas, who discovered double-stranded RNA could lead to gene silencing in C. elegans. So C. elegans was a small animal which proved worth, which really proved a lot while working on this kind of this thing. It grows very fast, and we can really follow the pathways of these microRNAs when we extract them. So they are the ones who said that double-stranded RNAs are very important. And then we have this Andy Fire and Craig Mello, who coined the term RNAi. RNAi is RNA interference. So this interference RNAs is the one which will silence the mechanisms performed by these double-stranded RNAs. So now the research goes on, and we have the Tom Tuskegee and the other group in Max Planck, and they have shown about the gene-specific silencing in malignant cells. So this is how originates capped polyadenylated full-length precursor microRNA. How it is done is, they have a hairpin-like structure, and mature miRNAs have 22 nucleotides, and they were first discovered by Victor Ambrose, and they named it as LIN4, right? And then LET4 discovered by 2000 by Frank Slacks as a postdoc working at Harvard University. So I'll tell you how this is produced in the cell. The microRNAs are initiated through a process where they have the cap-like structure, it is a hairpin-like structure, and RNAs, two enzymes called DROSHA and PASHA. These two enzymes, they pick up these microRNAs, and then when they have it in the nucleus, then this is exported by another enzyme called exportin into the cytoplasm. In the cytoplasm, you have another RNase enzyme which captures this, and it removes the loop, and then you have this. After exporting, you have the pre-microRNA, and this forms the MIR duplex. So this duplex is again captured by the RISC assembly, and then it separates into 22 nucleotides, 2 strands of 22 nucleotides. Now this is the mature RNA, which plays important roles in current biology. Hello. So this is how we come to getting the microRNAs, which we now have to study what the role of this is going to be, the mature RNA, microRNA. So here what it does is it attacks the messenger RNA. So the messenger RNA, the polyadenylated binding proteins, it binds to the 3' region, and then it stops the RNA, ribosomal RNA, so that it doesn't make the protein. So it disrupts the microRNA, and then it degrades the whole thing. So it prevents the ribosomal RNA to do its job. So here, the key players in this process are the human drosha, and then agronaut proteins, and you have the double-stranded binding proteins, and you have the dead box helicases, which will capture all these molecules. Then, briefly about these players, this drosha and pasha, they are part of the microprocessor protein complex, which could be 600 to 650 kilodaltons. And drosha and dicer RNAs too is double-stranded binding protein, and exportin, which is a member of the karyofuranine, is the nucleocytoplasmic transportation factors that requires RAN and GTP for its function, and agronauts are the RNase helicases. So what we studied is these two players. This is microRNA 149, microRNA 196. All these names have been given in literature for various purposes. So we just have to select what is the function of this, what could be this thing. So in many cancers they have functions, but in oral cancer nobody has done the work on these. So that is why we took this, and then we wanted to see what their roles could be in oral cancers. Because both function as oncogenes and also as tumor suppressor genes. Why means these small microRNAs will silence the P10 and P16, which are tumor suppressor genes. So when tumor suppressor genes are suppressed, then definitely cancer is going to progress. So this is how these two genes are very important. And then they have this metabolic regulation of 375 MIR, that means insulin secretion is affected. And also multiple genomic loci in different expression patterns are affected. So here, what I want to tell you is these tiny microRNAs, 22 nucleotide longs, they bind to the 3' untranslated region of messenger RNAs, and then they start degrading resulting in the translation repression or transcript degradation. So at least 2,500 RNAs have been discovered, and they have shown the process how this happens. And they are present in about 30 to 50% of the genes that code for these proteins. They are controlled by miRNAs. So many genes are controlled by these tiny 22 nucleotide miRNAs, which is being discovered for the first time, because we didn't know that there are so many players in the cancer, that this is one of the important players which was overlooked all these years. So these have emerged as integral components of various biological processes. Not only that, they include cell proliferation and also migration, differentiation, apoptosis, and also angiogenesis. So here it has been demonstrated that any altered expression of this is associated with tumorigenesis. And by regulating multiple potential target genes, MIR expression may lead to pathological changes in cells, ultimately controlling the development of cancer. So our study, we wanted to study what are the roles of these in oral cancers. So there are various reports in literature that they have been playing an important role in oral cancers. So we looked at that, and as you know, oral cancer arises as a primary lesion originating in any of the tissues of the mouth, and they are rarely extension from neighboring anatomical structures such as nasal cavity, or they can metastasize to distant origin also. So you know already the causes and risk of these. Just to go through that, alcohol is one of the culprits leading to oral cancers. Cigarette is the most, tobacco and cigarette and all this is the risk factors for oral cancers. One of the greatest risk factors is tobacco and its consumption in various forms, and also viruses also, NPVs, Epstein virus, Bar virus, and so many kinds of viruses in the oral cancer also are responsible for causing oral cancers. So this is the tongue, and you can see and you can detect it, and here it is below the tongue. You can see this spot and detect it. So these are preventable cancers. However, they don't come to the doctors till they are in the third or the fourth stage, because they think that small source, they can cure it by themselves by applying some glycerin or whatever. So they treat themselves and they don't come to the oncologist or don't get it tested and don't get it diagnosed. So that is how a preventable cancer becomes cancer which leads to mortality. So this role of microRNAs. So we selected the microRNAs, a few, 2 of them for polymorphic studies, SNP studies, and about 3 or 4 for expression studies. And then we selected some which are tumor suppressor genes, identification of their role in oral squamous cell carcinoma. We correlated all this with clinical and pathological conditions using statistical analysis. So these are the ones which we selected. Why we selected MIR 142 and 196A2? Because they play a role in P10 and P16 suppression. So the progress of cancer is taking place, and it leads to carcinogenesis and aggressive cancers. So they are very important microRNAs. If we identify these are very important biomarkers. So many therapeutics, we can target those for therapeutic responses. So the study design, we obtained the cancer samples from a cancer hospital, and then we explained to the participants about what we would like to do with them, with the biopsies. And then these are the patient profiles, where we have so many males, females, and the age group where this cancer is prevalent was 46 to 65, where we have 62% of these and controls 58. And we have classified how many were alcoholic, smokers and chewers, and how many had cancer of the tongue or buccal mucosa or mandible and in the oral cavity. And the stages in which we found these patients are, 50% of them having come to us at stage 3, and next will be the stage 4, stage 2 and stage 1 are very few people who come to the oncologist for treatments. So for sample collection, we had the blood samples for doing the SNP studies. We had the biopsy samples for doing the microRNA expression studies. Then we looked at the experimental details. All these details have been already published in my papers, which are available. And microRNA gene polymorphisms, which we did for microRNA 149. Why this is so important is because it can serve as an oncogene and a tumor suppressor gene. like I told you, it suppresses the PTEN properties, and it leads to, in most of the cancers such as head and neck cancer, lung cancer, gastric cancer, it has been described that 149 MIR plays a very important role. So in oral cancers, what we found was it was down-regulated. Now looking at the SNPs, we have genotyped this, and we find this is the wild type, which is very important, and without this, there will be without this genotype, the cells cannot survive. So we have the homogeneous and heterogeneous, and also we have the mutated, and also the double mutations with the CC and CT. All this has been done by looking at the PCR studies and looking at the sequencing studies, and we could get this information. And then genotypes were fully associated. We classified all these genotypes with reference to the patients. So in smokers, how many of these were CC genotypes, how many were there in TT genotypes, and how many were there as CT genotypes. So all this personal history and buccal mucosa and staging. So all these which are in red shows us that smokers and alcoholics are very prone to have these genotypes, and these genotypes can be biomarkers for diagnosis of cancer. So we looked at the 196A2. This study also has a crucial role in the development of cancer, because screening of patients for this proves clinically useful for prediction, that means for prognosis and for prevention of cancer. So this biomarker has been also very useful to look at and to classify them according to the genotypes, and then correlating this with the phenotypes. The phenotypes means clinically relating it to these are the phenotypes and these are the genotypes. So the genotype related to the phenotypes, you get very good information, and you can predict which is the one. MIR 196A2 will be useful as a prognostic marker for oral cancers. So here we have the results of SNPs, microRNAs. So that means we found that they are interfering with messenger RNA, and our work highlighted that 149 and 196 single nucleotide polymorphisms is significantly associated with the phenotypes of the patients. So here now we look at some expression studies. What happens is we look at MIR21, MIR137, MIR200C. All these MIRs we had to pick up from the available literature in other cancers, and to see if they are playing such important roles in oral cancer. Evergreen assay, and using qPCR, RT-PCR, and then the technology, all these steps are followed, and how we did all this is present in publications which I can show you, and I need not read out the complete protocol of the experimental protocol, which is take a long time. Only this CT images is where we find the important, the melting curve of all these 4 microRNAs. So with using that data, we can say whether their expression is altered or not. So in all the 4 MIR21, we found significant altered expressions. So here we looked at the p-values. Here also we have altered expressions, but sometimes the p-value doesn't show that it is very significant. But overall, you can find that the significance of these microRNAs, 200C, you can show that it is the altered expression is very significant in such case, in oral cancer cases. So the decreased tumor expression of microRNAs was reported by other authors also, but in other cancers. They were associated with poor survival in patients, especially with brain cancer, pancreas cancer, and ovarian cancers. We also found the down expression in oral cancers. So first time we are reporting this work in oral cancers. Now looking at MIR205, again we found a very significant association due to the alterations of these microRNAs. So these altered expressions may be associated with cancer initiation or progression and metastatic capabilities of the cancer cells. So microRNA 205 seems to be one of the key regulators in gene expression in endometrial cancers also. So here MIR205 may be key factor in determining tumor initiation, progression and cancer invasions. So here its exact molecular way of action has not been elucidated. Its role in malignancy remains controversial, as MIR205 may act as tumor suppressor or as an oncogene depending on which type of cancer it is found. So MIR205 can also modulate epithelial to mesenchymal transition, that is the EMT transition it is called. So this has mainly by E-cathedrin, which regulates via ZEB1 and ZEB2 inhibition, and also MIR205-5P can be negatively correlated by HER2, possibly contributing for the worst prognosis associated with HER2 enriched subtypes, mostly in breast cancers. So here microRNAs was revealed as an attractive prognostic tumor biomarker in recent studies. So looking at these, we can say that this can be definitely used to predict whether oral cancer is going to be aggressive in those patients. So this is a wonderful biomarker to be available to work with. So this biomarker has been identified by us as it's going to be an attractive prognostic marker in human cancers, in oral cancers. So microRNA 205 also specifically activates the tumor suppressor genes like IL-24 and IL-32 by targeting specific sites. Therefore, we need to look at the promoters which participates in the epithelial mesenchymal transition regulating zinc finger EVOX, ZEB1 and ZEB2. So to look at the altered profiles, we have the stage 1, stage 2, stage 3, stage 4. So MIR21 is highly expressed in stage 3, whereas MIR137 is highly expressed at the stage 2. So when we can look at MIR136 expression in patients at an early stage, it will be wonderful that they can be treated better and not wait till they come to stage 3 or stage 4. So the other two are expressed in stage 3 and stage 4. So here talking about the occurrence of these, altered expression of miRNAs according to their location. So here you have the tongue is in blue color and buccal mucosa in the red. So in buccal mucosa it's highly expressed, and in oral cavity also it's highly expressed, whereas it is very low expressions in mandibles. So these are the profiles in the various parts of the miRNA expressions. So here we have our experiments. We have identified upregulation of this MIR21, and we have the other three downregulated. So we have the expression of MIR137, 200 and 205 we found to be downregulated. So only one microRNA was upregulated. However, that also is very important because it is a biomarker and target for cancer treatments likely to improve the outcome of patients with cancers. So this interferes with the therapeutic agents while treating the cancer. So this will be a very important diagnostic marker during therapeutics. Whereas these down-regulated are very important biomarkers for looking at the cancer diagnosis and prognosis. So, for example, MIR 200 has a significant role in the initiation, progression, metastasis of many ovarian cancers, and also it has been studied as a tumor suppressor genes in this EMT for the expression of E-cathepsin, the classical calcium-dependent cell-to-cell adhesion protein to maintain the epithelial cell phenotypes. So the members of also this 200, they have been down-regulated in many tumors, and they are thus inhibiting migration and tumor cell adhesions, and also transition from epithelial to mesenchymal transition, and also angiogenesis. So the importance of these microRNAs and their expression levels tells us that MIR 200 is a family which inhibits the EMT and maintains epithelial phenotype by directly targeting the transcriptional repressor of E-cathepsin. And also, to conclude, I can say that it is from this investigation that microRNAs have important role in cancer development, and we have uncovered a few mechanisms of miRNAs, microRNAs regulation and DNA methylation in oral cancer etiology, which has been less explored area. Therefore, overexpression of MIR21, microRNA 21 in patients diagnosed with oral squamous cell carcinoma was found to alter the methylation status of the molecule, which is P10, which is a tumor suppressor gene. Then we go to the polymorphisms of MIR 149 and 162, 196 genes. They were found to influence gene expression in all the tumor suppressor genes, and also the mechanism by which the polymorphism affects gene expression include variables in the cell, which has been explored to some extent by our results which have been published, and the investigation has proved for the first time that there exists a strong association between MIR 149 and MIR 196 in oral squamous cell carcinomas. Hence, we say that these microRNAs can be useful biomarkers. So, cancer metastasis accounts for almost all cancer-related deaths. And metastasis is a multi-step process, and it involves tumor cell dissemination from the primary tumor colonizing to distant places. And then the improved ability of tumor cells to undergo migration and invasion contributes the first steps to this process. And these are the microRNAs, which are the targets. MIR 149 was significantly downregulated, therefore promoting metastasis in cancer cells. So these are some of the mechanisms which we unfold while we are looking detailed into the cancer microorganisms, microRNAs, and therefore the cancer resistance to chemotherapy we can find out by looking at the upregulation and downregulation of MIR21, and also studies have shown that MIR 149 also participates in regulating drug sensitivity in cancers. So these areas have not been explored earlier, except now the literature is mounting and people are working more on these tiny molecules known as microRNAs, and we know that the players, the MIR-149 plays important role in tumorigenesis, and also there are a large number of studies exploring the effect of MIR-149 as therapeutic targets. So these are important findings in the area of microRNAs. So the expression of this microRNA, which is 149, was significantly lower in our tissues when compared to normal tissues, but they had a very important role and they improved the survival once it is targeted, and then the duration and lower mortality risks were also observed using Kaplan-Meier and multivariate analysis. So it was further reported that this pathway, AKT and mTOR pathways, was hyperactive when there is lower expression of MIR-149. So such pathway studies and how these MIR will target which pathway in the cancer cells is very important to unveil, so that we can understand the role of MIRs and how they target the cancer cells. The mechanisms have to be unfolded by looking at these signaling pathways of these MIRs. Other studies have also shown that MIRs in the tumorigenesis of various types of cancers have been reported to be involved in numerous cell processes, including apoptosis, proliferation, migration, and thus suggesting MIRs have an important role in cancer progression. So, our evidence points out that the benefits, particularly using miRNAs as cancer biomarkers. All our evidence says that these are important biomarkers for cancers. Then the relative expression of 4 microRNAs, which we did, was MIR-21, 137, 200 and 205. The process from primary transcript is dynamically regulated. So these findings of ours, they are reported by doing this quantitative RT-PCR studies in our South Indian populations. So we found that the upregulated microRNAs targeted gene enriched in this pathway, PI3K, that is PI3K kinases and AKT pathways. This is the signaling pathways through which they are activated or they are downregulated, and therefore they inhibit these pathways so that they lead to evasion of apoptosis. So when there is no apoptosis, there is more increase of defective cells which will propagate and form cancers. So this is very important, and in these cells and cell cycle checkpoints also. So they bypass all this so that the defective cells will propagate and continue carcinogenic process. So these are the mechanisms which we found while working with miRNAs in the 4 expression studies of those miRNA expression studies, which included that 137, 200 and 205. Out of these PI3 kinase and AKT signaling pathways are being, through that, they are being targeted. So these alterations in the cellular level of microRNAs, they have impact on the proteome of cancer cell therapy, contributing to oral carcinogenesis. So high expression of miRNA-21 may be characteristics of cancer cells, and they represent a common feature of pathological cell growth in cell growth or cell stress. So we have the results that miRNAs are extensively involved in cancer pathogenesis of solid tumors and support their functions as either dominant or recessive cancer genes. So overall, we have this center spread where we have the oral cancer and how the miRNA upregulation increases the suppression of PTEN and through the PIK3 pathway, which leads to the AKT signaling pathway, suppresses and allows and do not allow apoptosis pathways, whereas the MIR149 and MIR196A2 will interfere through this process, and they have the tumor invasion and metastasis, which promotes by these microRNA down regulations. This novel approach to cancer biology, therefore, I can say it has immense potential to advance cancer diagnosis and stratification. And also, it is the first comprehensive study to explore the role of a few microRNAs in oral cancer in our South Indian population, and the accessibility of oral cavity for microRNAs makes an attractive target for gene silencing strategies. One important property of gene polymorphism is that their incidence can vary substantially between different racial or ethnic populations. So what can be true for maybe South Indian population of the India may not be applicable to the population in Mexico. So, or therefore, targeting miRNAs is an attractive, important anti-cancer strategy, and also not all cancers can be treated by the same microRNA based strategy. However, comprehensive understanding of the microRNA regulatory mechanism is also necessary to improve the design of microRNA based therapeutics. And also there is a need for safe and efficient delivery systems when using this approach as for anti-cancer treatments. So this is what, but this is only a precaution. So these small non-coding RNA transcripts are likely to outnumber the group of protein coding sequences, and they hold very good promise for new discoveries and mechanistic explanations for essential biological phenomena and pathologies. So best characterized coding RNA family consists in the humans of about 1,400 microRNAs, for which abundant evidence is also available, and they demonstrate the importance of that in normal development and differentiation, and also in human diseases such as cancers. So in this summary, we summarize the concepts concerning the involvement of microRNA in oral cancers, and we have shown the regulation of the key cancer-related pathways like mTOR and also the PIK3 kinase and AKT signaling pathways. So which are the ones which are related to the microRNAs and their gene silencing technologies. So importantly, microRNA molecules are already entering the clinic as diagnostic and prognostic biomarkers for patient stratification and also as therapeutic agent for cancers. So this is the most important discoveries and the latest trends in cancer research. So I thank everybody, and I thank my organizers, Bhagwan Mahavir Medical Research Center, my oncologist, and my students who have been running around the hospitals to get me the samples, biopsy samples, and through them I could carry out a lot of this work, and we have still some more data to be published, and we are still working into this. I still have 4 PhD students who are sincerely working with me and helping us to discover more and more novel processes for cancer treatment and for technology. So, thank you. Any questions?
Wonderful. I'll have one. firstly, we must thank you for this wonderful presentation and very valuable and very important study, research material. This is priceless. Very good. Just one question. mRNAs, as you know, they come from the same genus of iRNAs. Roots are the same. I think, let's say, coming from, from
the
right, yeah, the smaller but new. I mean, let's say internuclear later generations of the same iRNAs. Now, depending upon the circumstances, internuclearly, they can act as tumor suppressors as well as, very interestingly, oncogenes. Now, let's say, for example, 137,
yes.
200, 205
yes.
MIRs,
yes.
and 21.
Yes.
Now, have you ever had a plan to document the circumstances, or have you ever planned to list the documented circumstances of suppressor and also at the same time as oncogenes, acting as oncogenes. Have you ever had any documentation of that?
Yes.
I mean, under which circumstances, internuclear circumstances?
One is downregulated
Right.
and 3 are upregulated.
Right.
So
yeah, this is what I mean.
Yes.
Yeah.
So one which is upregulated, when it has its expression more,
Right.
and it suppresses through the mTOR pathway, the P10
Right.
and the P16.
Yeah.
So through the cascade of events, it will suppress the other genes which are tumor suppressors.
Okay.
So then it becomes important, because then again it becomes that it promotes the tumorigenesis.
Right.
Even when they are down-regulated, they have the same, but through another pathway, signaling pathways.
Right.
So they also suppress, and they increase metastasis.
Mm-hmm.
So
right.
yes, and they also increase drug resistance to the tumors.
Mm-hmm. Right.
So these are also in a different way, but they promote the tumorigenesis process.
Mm-hmm.
So whether they are upregulated or downregulated, we need to know through which signaling pathway they are acting. So
Exactly.
they have different pathways,
Precisely.
but they are promoting cancer.
Right.
So one is, even though it is upregulated, it uses the P10 and P16 to stop the tumor suppressor, and it allows the cancer to progress.
Oh, yeah.
And whereas the other 3, they are down-regulated, and then they will promote the metastasis, they will promote the therapeutic agents to become resistant. So they have 2 different types of mechanisms by which these are acting. So these are very important microRNAs which we selected to see how important they could be in oral cancers, and they could be biomarkers of the oral cancers. So
so that is it.
Yeah.
So it has already been in process,
Yes.
my query. So it's already there. So it's so good to hear. Good to hear. So
Okay.
wonderful. Okay, thank you.
Okay.
Thank you very much. And